Great. Hello, everyone, and welcome to our final VanBook webinar of the season. We would like to begin by going over our schedule. We will start off with a few announcements, and then we'll go on to our introductory student talk, followed by our featured speaker, and then a question and answer period. However, for the student talk, please hold your questions till the end. But for our primary presentation, you are more than welcome to kind of engage and interact throughout the experience and ask your questions as we go along. We'd also like to take a moment to thank our sponsors. We have the SFU and UBC Bioinformatics graduate program, Genome BC, Ecoscope BC, the bioinformatics.ca, and particularly the Canadian Bioinformatics workshops, Abcelera, a local Vancouver operation, Langara College, as well as the ISCB and the Mon bug, that being the Montreal bug, and the Toronto bug, Tor bug. Oh, sorry, it suddenly changed my screen. There we go. All right, and also, seeing this is the end of our season, we are looking for suggestions for speakers for our next season coming up in September. So if you have anyone in mind that you would like to have us present, please feel free to go to vanbug.org and use the suggest a speaker option. Now, to begin with our student talk, we have Fatih Karalanoglu from Dr. Faraz Hash's lab at UBC, and he will be presenting today on GenION, an accurate tool to detect gene fusion from long transcriptomic reads. Followed by our main presentation, and we'd like to welcome Dr. Jeff. He is an associate professor of parasitology and the Department of Animal Science at the University of McGill. He is also a Canadian Research Chair in Bioinformatics and Big Data Analytics. His research explores innovative and practical ways to address current challenges in big data analytics that arise from biomedical and environmental research, metabolomics, transcriptomics, microbiomics, and multiomics integrations. His team has developed many tools used by researchers worldwide, and they are actively developing a new generation computational framework that integrates cloud computing, machine learning, and visual analytics to enable high-throughput data analysis. He's authored over 75 journal publications, 8 book chapters, and has been ranked a global highly cited researcher since 2019. He has over 20,000 citations and an H-index of 41. So we look forward to his presentation on towards principled approaches for multi-omics integration. And all that being said, we'll move forward. And Fatih, you are more than welcome to share your screen.
Yeah, 1 second. I think you need to exit first. Okay, thank you. Okay, can you see?
Yes, great, thank you.
Okay, hello, I'm Fatih, and I will present you my work on gene fusion discovery from long-read transcriptomics data. So, gene fusions are chimeric genes formed by structural variations. These might be deletions, inversions, translocations, or duplications. For example, if a deletion between gene A and gene B happens, we will have a fusion gene containing this sequence from both genes. At first look, finding gene fusions from long-read transcriptomics data may look very simple. We map the long reads to reference genome or transcriptome, find reads that map to multiple genes, and call the gene fusions by clustering those genes. Quickly after working in this field, you see that this is very naive, and it is simply not possible to call the fusions like this. While the basic idea is there, we need to do more to accurately call gene fusions. For example, in the PEC biosequencing of MCF7 cell line, out of 2.4 million reads, there are 1,800,000 reads that are mapped to multiple genes. And only 2,000 of these are coming from high confidence candidates. So what are the remaining reads, where are the remaining reads coming from, is the question. We classify the chimeric reads, RNA reads in your sequencing data into 3 categories: the gene fusions, read-throughs, and random pairings. The gene fusions and read-throughs actually exist in the cell, but the random pairings, we suspect that they are created during sequencing or library preparation. Read-throughs are similar to gene fusions, but they are only formed between the genes that are on the same strand and they are in the same proximity. Of course, they are also in the same chromosome. But different than gene fusions, in these, for example, we don't have any structural variations between the genes. There are no deletions. The transcription simply does not stop at the end of the first gene and continues transcribing the exons from the second gene. Random pairings are a bit different. So the molecules from different genes are randomly attached together during the sequencing. Since this is a random event, the count of such chimeras is correlated with the counts of the normal reads of the member genes with some constant pairing ratio. While this may be insignificant and most of these random pairings will only have 1 or 2 read support. But you might ask, what's the likelihood of consistently getting random pairings between the same gene pair? Well, we observed in the MCF7 cell line up to 150 reads from a gene pair, and without a smart filtering, this will look like a very strong gene fusion candidate. So we propose a Jianguo method, which is made of 3 main steps: the mapping, chimeric read identification, and clustering and characterization of the chimeric reads. The clustering is simply grouping together the chimeric reads that are likely to originate from the same molecule, and characterization is labeling these clusters as one of the 3 categories we mentioned earlier. So in the mapping part, to our knowledge, there are 2 mappers that work on the long RNA data, which are called spice-aware mappers, Minimap2 and Dassault. The problem with the Minimap2 is that it simply cannot map to smaller exons. While this is not a problem for the most gene fusions, if the head or tail gene of a fusion is short, Minimap will not be able to map it. What I'm meaning by short, for example, in the Tempus2 ERK fusion of which is very common in the prostate cancer, the Tempus2 part of the gene fusion is less than 80 base pairs. It is almost impossible to find these with the Minimap2. On the other hand, while the Dassault is good at mapping to shorter exons, it does not by default report split mappings. So if it will only find gene fusions if their genes are in close proximity and they are on the same strand. To overcome this problem, we map the reads first with Dassault and mask the map segments of these reads first, then map them again. So we have the split alignments if they exist. So we map both parts of the read. Sorry. Okay. So in the chimeric read identification, for each read, we have set of aligned segments from the mapper. And for each segment, we have a set of exons possibly coming from different genes or different isoforms of the same genes. We use a dynamic programming algorithm to find a chain of segment exon pairs where each segment is used only once and the overlaps between the segments and exons are maximized. We also penalize the consecutive segments belonging to different genes because we want to prioritize normal chains over chimeric genes. We don't want to get false positives because of multi-parameters. In the clustering, we group the reads supporting the same gene pair, which we discovered during the chaining part, and characterize its type using the exon structure, also discovered in the chaining part. and the two scoring schemes we developed in this work. The exon structure is mostly we follow the integrate paper. In this paper, they have shorties from whole transcriptome, also on whole genome, and they provide statistics about the likelihood of a chimeric gene fusion candidate having genomic evidence. So for this score, FIN means fusion inverse normal score. We simply divide the number of fusion reads by the number of reads from the individual genes, number of normal reads. The reasoning behind this is that structural events causing these gene fusions, we expect them also to reduce number of normal RNA reads coming from these genes. So with this score, we expect gene fusions having higher score than read-throughs, and read-throughs having higher score than random pairings, which we expect should be very similar to 0. And we also expect FAN of homozygous events to be higher than heterozygous ones. We also introduce FFIGF score, which is fusion frequency, inverse gene frequency. It may look similar, but the idea behind this is that any gene is expected to be at most part of a single gene fusion event, since it removes the possibility of having another gene fusion event on top of it. Like we can have two events if the genome is diploid, but having the same gene twice is very unlikely. So also the problem with the RNA data is that high support, having high number of reads supporting an event does not necessarily mean high confidence. Remember the example I gave with 150 read random pairing, because the number of reads is scaled by the expression of the genes. In this score, we penalize the number of reads by the other candidate gene fusions we find with this using the same genes, gene A, or for example, for a score of FFIG for A and B gene fusion, we also divide this by a number of gene fusions containing A or B separately. Results. So to test this, we created a simulation. We simulated the transcriptome using 2 RB1 prostate cancer cell line expression profile. We added 16 validated gene fusions from the Cosmic database. We also added 37 non-read-through chimeras. We generated reads with nanopore error signature using the simulator called BedRead, generating a total of almost 3 million reads, and only 3,000 of these are actually from the simulated gene fusions. After simulating the reads, we created random pairings with 1% ratio. We did this by shuffling the reads, then appending the consecutive reads together with the 1% chance. In this simulation, the genome was able to find 15 out of 16 simulations, and the only fusion we missed was this one, because the member genes of this fusion were overlapping, and we were filtering out the overlapping candidates. Here is the FFIGF and FAM values of the candidates we have. Each point is a candidate chimera, and the x dimension is the FIN score, and y dimension is the FFIGF score. Here is the red points are filtered because they have low number of reads, which are the random pairings having less than 3 support. These are very easy to filter out even without our method. The problem starts with the blue ones. Blue ones actually have higher number of supports, which are not possible to filter out using the read count. But they have very low FIN score because of this. Because of the random chance being very low between the random pairing happening. So this score allows us to differentiate between actual gene fusions and read-throughs and the random pairings in this sense. We can just find the point and cut the lower candidates with lower FAN at that point. And in the right end of the screen, we see the simulation, and you can see that the the profile is very similar to the real data, which supports our claim that the random pairings actually happen as we expect them to be. The only issue here, you can see the biggest issue, you can see that the FAN scores in the simulations are a bit higher than the real data, which is mostly caused by the homozygous events being rare in the real data. Also, the mismappings, the missing mappings causing the normal expressions to be higher than normal. That was all for my presentation. Thank you for listening. I thank my supervisors for their help and my research group. Thank you.
Great, thank you so much, Fatih. That was really interesting. If anyone has any questions, please feel free to unmute yourself, or if you would rather, you can also type it in the chat and myself or another moderator will read it out for Fatih to respond to.
Maybe I'll ask one, obviously. So whole thing is done on RNA-seq data, right? And we claim still that the fusion are a genomic event, right? Do we need the genomic data to, orthogonally validate this fusion, or can we just rely on the scoring coming from this?
Well, just relying on the score needs, I think, more experiments. But the idea is that finally we shouldn't need genomic data to do this. That would just break the point. Like in the ideal situation, we should not be required to do it, I would say.
Okay, thanks.
Okay, well, looks like there's no more questions, so I guess we'll move on. Thank you so much.
Thank you.
And when you're ready, Jeff, please feel free to share your screen, and everyone, let's give a virtual welcome to Dr. Jeff and his presentation on his team's journey navigating multi-omic studies.
Hello, everyone. Can you see my screen?
Yep.
Okay. So, oh, good morning for you guys. I'm afternoon already, and it's my pleasure to present. So, let me introduce myself, because I'm not I've been in Vancouver for 2.5 years. So I did my PhD in University of Alberta. Then I drove across the Rocky Mountains to UBC at the big Department of Microbiology Immunology for 2.5 years. So my PhD was on metabolomics, and my postdoc is mainly on RNA-seq analysis. And I did attend several VANBACs during that time, but it's traveled quite a bit. And so I'm very happy and feel comfortable to go back and see VANBAC. And here at McGill, I started as an assistant prof about 5 years ago as in a faculty of agriculture and environmental. So it's very quiet, peaceful place. And you can see a lot of green things going on. So especially it's such a blessing during COVID. So we can more or less normal. And you see this map, it's a very long way from Vancouver, but it's quite a nice journey. And I didn't drove, but overall, it's, I feel quite connected still. So here is at McGill, and what I'm doing since 2015 is that because I had a lot of training in metabolomics and RNA-seq, so we are expanding from microbiome data And also this faculty have a lot of different other data. So overall is that we just talking on big data and actually very, how to say, a very overlap with what's going on in the whole wild world. So big data analytics, and how do we integrate data? What's the best way to do? So you can see there's the key terms usually summarize my research. So one of them is data integration, one is called visual analytics. So visual analytics, one of the approaches we want to let people to understanding complex data. So I will gradually introduce some of the how to say, some of the presentations we try to showcase how it works. On the right-hand side is a mass spec. So I'm not purely doing computing. I also do mass spec because I love it, and it seems quite interesting. You can do some predictions. You can also validate in the lab. So it's a quite a different word, I will say, because I'm crossing both and back and forth. I realized that the mentality is very different. Sometimes you can get very high and dry, didn't know what's reality. Reality is quite a different situation. And we just refine one single protocol and take several months, but we can do the simulation on the big computer overnight. So you have to adjust your pace and patience to get things coordinated from computation and analytical. So I think everyone is working on the same page, like here defined about 10 years ago. The big context, so we sequence the human genome and in the turn of this century, and we want to understand biology in the first 10 years. And now we want to understanding the phenotypes, including disease, a lot of things. So in order to do that, we gradually introduce more dimensions from like RNA-seq to epigenomics and metabolomics, microbiome, and a variety of other things, single omics stuff. So it's really a booming blessing for people who is more computationally strong. And for me is that I found it's quite empowered by you see that so many data, and the data can help you inform your decisions. And so overall is that so for the beyond 2020 here is that we want to translate what we learn from data into actions, into insight, and to really have an impact. So this is something a lot of my findings and proposals try to aim into to reach. So this is very hard. A lot of times we found a very cool algorithms and working very well, but nobody use it. And sometimes you feel it's very frustrated. Why things like that? Because a lot of us, we don't actual users. Okay, we developing tools or developing algorithm, put it there, and use or not, we don't have too much control. So this is sometimes we need to promote the work and have a kind of more translational activities to really push down. And it's the real people in the front line who make the changes. So if you can convince them to use your tools, and it will have a huge impact. I can clearly see a lot of very cool development in the field, and I've evaluated it's so good, and it just takes quite a long, sometimes being ignored. So this is sometimes I'm just talking about the bioinformatics. We need to more translational bioinformatics. So there's need to be a more targeted translational perspective for this. So I will gradually introduce some of my approaches, actually go all the way to the end, to the users. But let's go back to the multi-omics, which is focus today, is because we want to understand this complex disease, including cancer just presented. And there's so many things changed. And naturally, we need to marry multiple layers of things, including genomics, transcriptomics, proteomics, metabolomics. So we do get a lot of data, and we do want to understand it. So here's one thing is that the big data, we're collecting a lot. And we would like to get to a system on biology, understand everything. But most times we get, patterns and chance. And so it always need to be connected with whatever you are studying. So this is something that it's not automated to tell you everything. It's always your data and your context. So these two things meant to be connected to make it more meaningful. So I will gradually give some more point on that direction. So here's that if we have this a lot of data and what we want to do with it. So in this in the last 5 years since I came to McGill, and we gradually adopt two approaches. Of course, this is a very natural approaches, but realized in the after a few years, and it's quite interesting. So there's two things: we get big data, and it's very large. Okay, we get hundreds of gigabytes of data. Really, getting data processed in a fast enough, and even if you get a high performance server, it takes sometime overnight, or sometime if you're doing simulations or stuff, it takes longer than that. And after you get your data, you do get your information. That's usually a table, and usually a high dimensional table, and it's much smaller, so you can share with your colleague and doing data analysis. So this is where actual data integration happens here. We don't do the data integration from the raw data. Usually we do integration from the profile data. Usually it's such a table. So when we have such a table, and we just, sometimes we think about biology, sometimes if you don't think about biology, just think about data. And that table, how we can connect in this different table together. There's a lot of things beyond bioinformatics. There's a lot of things going on with machine learning and AI. So they need to deal with a lot of data forms like this. So if we think about these data properties and how do we deal with them, and there's two different approaches. For the raw data, we usually don't move them around, and we have a local server, and so you just produce the data and you process the data, just write beside your sequencer. For in my case, we just be right beside the mass spec. So in the same room, connected with high-speed internet. So after we're doing that, and we were going to process the raw data into a data matrix which I shared with just in the previous slides. It's much smaller. And how we deal with that data, we usually upload to the cloud, and sometimes private, sometimes public, and usually is whoever designed the experiment, designed the study, they will analyze the data. So this is a very more customized, different data like metabolomics, RNA-seq. It's quite different procedures, but the raw data process more or less we can standardize, so we just run it in another way. So this is two different approaches. We have a local and a cloud-based approach. So one of the a lot of tools we developed actually just to use this concept. We have the local cluster, and which is processing raw data, and we have the galaxies. We have other workflows we developed recently, and then we use a public cloud, and this is mainly we use Google, and we also developed approaches I mentioned at the beginning called visual analytics. So why the visual analytics are critical in the application is that a lot of times you want to communicate it to the researchers who's actually making a decisions, and they need to understand data very well. You cannot just give them a few values and tell them, believe this. This is the most important things you should pay attention to. A lot of times, especially for the omics and multi-omics, and they need to get more context. So a lot of time visualization is very important, and a lot of things you have to be communicated in a way that not overwhelmingly a lot, but also interactive, allow them to query the data. So a lot of times it's with this browser-based interactive, they spend quite a long time. So they spend days and nights and try to understand data. So this is sometimes when decision is making and data understanding on translation is coming. So this last part is that from the web browser to the end user, it's usually people don't quite think too much, but I just mentioned at the beginning is that it's quite meaningful when you try to think how to communicate the result to the users, which can make a difference. So I mentioned about the overall big data framework. And now before I move on to multi-omics, I would like to emphasize the single omics, which is the basics and still the most critical part before we can understand multi-omics. So every single omics and I'm mainly referring to the functional genomics from downstream of genomics. So maybe genomics slightly different, but here it applies to transcriptomics, proteomics, metabolomics, pretty sure, and even include microbiome. And we can use almost a standardized workflow to analyze them, all of them. So at the beginning, we need to do the QC and quality assurance and make sure they are proper, no outlier, no strong, strangest behaviors. And then we can do some pattern detections or statistical analysis. So this is usually, we talk about this basic t-test, ANOVA, clustering analysis, and the classification, machine learning stuff. It's all common. So once we get a matrix, we can do a lot of things disregarding which omics. And usually for this well-established omics, and we can do further functional interpretation, like pathway analysis, enrichment analysis. And this is quite common. And now I think it's well established. A lot of researchers are very comfortable. They can do their RNA-seq analysis all by themselves using some tools. So this is last 10 years, our effort is really paid. So, and the single omics is really well established. A lot of people can do it, no problem. And finally, there's some unique things. For example, genomics or metabolomics have some things a bit unique, but the majority they shared. So single omics is still the basic for multi-omics. And why I'm saying this is because we need to understand our single omics as thoroughly as possible before we move on to multi-omics. The investment of multi-omics is usually high, and so we need to really do our homework before we integrate them. So I introduced several of the approaches on the single omics and the big data framework. And actually we developed the whole pipelines for the common omics we usually see. And here's I just show these tools quite used a lot. This is already outdated and probably doubled since last year for a lot of users. So we have metabolomics, we have transcriptomics, which is called network analyst, but it's mainly for gene expression data analysis, and microbiome analyst, which is for microbiome data analysis. So it's used quite a lot, and so a lot of time is we just put our tools to the cloud, and people upload their matrix and try to understand what's going on. And some stats is different, but a lot of data pre-processing is quite shared. So it is very useful. And now is that we get all the omics, and the people always want to get actual more. And now the cost is low, and people usually do more omics. So last several years, I get more queries about how I get RNA-seq, I also get metabolomics, how should I integrate them? And a lot of times it's actually quite natural. What I'm saying is that you get your basics done and you get extra things and just add it on and think more, and the story will be fuller, will be more enriched. So this is something it's quite a, how to say it, as a natural transition from single omics to multi-omics. And clearly that a lot of approaches not as standardized as I showed you before for single omics. the multiomics is still, I'm not saying a mess, but it's quite a diverse. So all different approaches being tried, and there's no, how to say, a convergence very clear, and based on my reading. So definitely it's getting better and better, but so far it's not a clear path there yet. But today I will try to say something during my own experience. I also feel it's quite easy to follow. Okay, and a lot here is that I want to say is that there's a lot of approaches that you can pick, but most time you should focus on is how do you do the analysis. Is need to be transparent and interpretable result. So that a lot of times you have multi-data. How do you making sense of data and putting a lot of complex approaches together? That naturally means the result became obvious. It sometimes making it worse. So that's the overall message I want to get. So here is that multiomics, what we should expect from multiomics. I already shared a bit at the beginning, and people equate multiomics to system biology, which I think is inappropriate, especially at this stage. We get a lot of, we get multiomics, but we don't necessarily get a system level understanding that close, because a lot of omics and not, how to say, it's not mature, the coverage is not good, and resolution is not high. So if we adjust our expectation, so multi-omics just more than a single omics. So you just focus on omics, one omics you get most, and gradually adding more, and you will find it's very natural to increase the values of what you have. But if you want to get more system biology, have that very high expectation, and you will be disappointed. So far that all the multi-omics analysis, and disregarding how fancy it is, and it's not very clear you get a whole system understanding. It's very rare, and once you get it, it's very how to say, high impact journal publication. But most time we should get much better patterns and chance. So this is something I would want to say is multi-omics. We still need to focus on the rich stories, more refined patterns, and stronger trends. This is something, if you focus on this, and multi-omics actually quite easy to do. So if we put our focus perspective in the right place, and what different approaches we can do, and I just come up with 4 general conceptual path. Of course, you can refine them, get 5 or 6 or even to 10, but it will be hard to follow. And I just talking about this 4, and clearly I'm not saying this is very exhaustive list. But this 4 is actually quite natural. I hope you share with my going. So the first one, the single omics driven incremental integration. So this is what I already mentioned before, is it's very natural transference where you get more data and you're just expanding from what you already have and gradually increase them. So this is something most of us actually doing all the time. And this is not kind of, you don't need to rely on fancy tools. You just do it naturally. And the other one is correlation analysis. This is you get multi-data, you want to see how the actual connections. And the third one is we're talking about knowledge-driven integration. This is a very, how to say, a very booming, because especially for the human and model species, we get a lot of data gradually distilled into knowledge. So actually we can map our data to our knowledge. We get quite a lot of information already. And the last one is most actively, how to say, actively under development. A lot of the complex machine learning, multivariate statistics focus on the as a model data-driven integration. So they want to have more data and naturally come up to insight. So this one is have a lot of the publications, but usually is very far away from most of the researchers, because it's very complex. A lot of times it's a dark black box. But a lot of researchers result from these approaches became more kind of have a lot of success stories. And I will share some of them we used and feel it's quite useful. And we don't need to understand it deep how it works, because they are not designed to be transparent, but they show their performance actually very robust and to be trusted. So I'm focusing on the first one, is single omics driven incremental integration. So the first, this is if we just doing RNA-seq, we want to do metabolomics, and what we need to do. And the first thing is that we're going to do a single omics analysis. We try to understand the RNA-seq as much as possible, and we try to understand metabolomics as much as possible. Then we think how they conform each other or unrelated, or how they actually connected. So this is something is quite natural, and it's most useful and flexible. And I hope most time is actually you get extra information. Some is unique, some is actually shared and supporting. So you get your stories become more stronger. If it's really conflicting, you need to think about what's going on. Is at the same time frame? Sometimes, for example, metabolomics, RNA-seq, or proteomics, you need to see, when you're collecting samples and stuff, they are not happen at the same time domain. So you may not get the strong signals or correlated signals. So in that case, it happens at the study design. Okay, the data is collected, so you need to reconcile the data. And so this is something it's very critical, and actually most powerful is design the data, design a study, and collect the data, and think about the whole biology. Once you get data, you can actually put all things together and become a beautiful paper. So using very fancy algorithm at the very beginning is should not be encouraged. So always is study from very simple, and study from your biology, study from your design, and see the data, whether it's conform or not. So this is something I definitely encourage my students, and a lot of I have the collaborators try their best to think and read the background, what they have in their mind, and then once you get the data, and everything linked together, and a lot of the collaborators, they just so excited to say data. Oh, this making sense. This making sense. They put all the things together. Then that's something. Sometimes you see that things happens. So that means they thinking it quite a lot. They accumulate a lot of ideas in the brain, and so they are very natural for them. But for the people who are more junior in the field, and this require a lot of reading and connecting. So this happens in our brain, but our brain is very powerful. So it really, this step cannot be skipped. So if we try our best to at this stage, we can write a beautiful paper. Okay, a lot of times we use fancy tools, and we don't know what it does. So we only know the names and parameters. We couldn't even write a good paper. So this step is definitely need to try. We cannot skip this. Multiomics, starting from single omics, and we try our best. So now what's next? So we do our try to understand individual omics, but this is we try to understanding connecting them using our brain. But we can do some very simple statistical correlation analysis. Okay. So this is the second step I would like to recommend is correlation analysis. And here I showcase the two omics data. And they have features, high dimensional features. And usually each omics have more than 10,000 features. It's a lot. And there's definitely concerns about it's a lot how you correlate them. You get so many connections. And most times we just do things that we feel is reasonable. Is we do identify features that that are significant. Okay, this is not necessarily a differential expression analysis. It could be clustering analysis. It could be something else. And so there's many different approaches to identify the features that are significant or that are important to your needs. Now you can try to correlate these features from two different omics domains, and then you can explore their correlations within a beautiful heat map or networks. So this is very effective, and a lot of times it's very insightful. And I would like to see here is that how to select the features, how to compute the correlations. Each one almost have 10 different methods. So I don't cover and discuss them, but I think that you should try. And usually such approach is quite easy to follow. If you approach in this way and discuss how you get the result, and reviewers or your supervisors should be very clear to follow your thoughts, and clear. And at this stage, and you can show your result as a network, like here I showed you, and this is generated by one of our tools, and from two omics, and we can almost see the patterns, strong patterns, and on the right is I think it's proteomics and metabolomics. So their correlations and different color, including indicating what's positive, what's negative. And you can see a major cluster here, and they are really in pathways that are tightly correlated. And on the left-hand side is some of the major players that I didn't label because we want to. The label became messy. So it's several key players in this network. So for some experienced researchers, they can usually think about what's going on and further reading literature back and forth and basically solve the integration problem. So here you can see is that even we start using some tools to help you, the story still need your active thinking and what's the context, what's disease, what's learned. So come up with fill all the small gaps, get the stories out. For correlation analysis, it's very easy to follow, very easy to do, and I recommend that. So the next one I want to talk about is knowledge-based integration. So this one is also no need of fancy stuff, but you need to have a very extensive knowledge base. And this one is fine if you study human, we study mice, and you can collect a lot of public data. For example, we have the metabolic reaction pathways of networks, and you can connect the genes, proteins, metabolites all together. And you can have signaling pathways, have transcriptional factors, microRNAs, genes. So all of them are very naturally connected. And if these types of molecules are related to your omics layers, and you can do the same thing. Basically, you do individual omics data analysis. You identify features of interest. So whatever your interest could be based on associated with the phenotypes or clustering or something else, and you map them to this knowledge universe. And then you see how they connected with regard to knowledge or no knowledge. So, so this is another thing is we show it in network, but this network is not a correlation. It's network connected by the knowledge. So on the right-hand side, we just illustrating one of the networks we generated. This is based on protein-protein interaction network. I think it's about 5,000 proteins included, and we have illustrating about 8 different things put it there. So this is not closely connected. We just show if you have your interest and how do you put it in a context, and you can map them to this network. And from network, you see how they connected and gradually come up with a more refined, maybe active subset of biological process. So once you have such a network, you can do a lot of analysis. And here I'm talking about the graph theories and topology analysis. You can also do some statistical permutation, see that some pathways enriched in certain subset of the seed modules. And also visualization wise, you can get a very good visualization, and for a few thousand, it's okay. And usually you can visualize, explore, see what's going on. So this is something we have a comprehensive knowledge base, and we can map individual features from individual omics and to this big knowledge base. Then we try to extract how they connected, what's the big story. So this is also easy to follow, easy to do, and hypothesis generation is good. So the only thing is that you need to pay attention is that if you don't have a high quality knowledge base, for example, if you study non-model species, it will be very hard to do such things. Also, is a novel connection will be hard because this is known. You're mapping to known. So if you found unknown novel things, it's not going to be easy. So this one should be used with the correlation network and try to complement each other. But what I want to say is most time is either if you have multi-omics data, either one will give you enough leads to generate your hypothesis, and the combination is natural, but sometimes you will get enough multi-omics. So this is one I mentioned about a very natural step from single omics and to correlation and to knowledge-based integration. And this is the last one, data model-driven approaches. So I don't pretend I'm an expert here, but I do actively follow what's going on. So this is one review published 2 years ago on nucleic acid research, and talking about multi-omics and multi-view approaches, computational approaches for integration. And they did a very good job. And for example, here they summarize how you actually integrate different omics. One approach is called early integration. Basically, you concatenate omics and put them together and do some clustering. And this one is, maybe you can do it, but I really doubt it will work well, just because different omics have different scale. Okay, concatenating even after you do a scaling adjustment usually doesn't working well. They have a very strong omics specific effect. So this concatenating omics usually should be very cautious. And what we usually do, we found useful is late integration, or so what people usually do is that do individual omics and try to come up with these correlations, then they finally connect them together. One of these approaches called the Similarity-Based Method, and compute per omic similarity and then do the connection. So this approach, one of them called a Multiview Clustering, I will show you in the coming slides. It's very natural. It's not happened from bioinformatics, but from the social media machine learning. And it's really seems useful. And I will introduce you in the next slides. But here is another thing I want to see is called dimension reduction. Dimension reduction, I think everybody is familiar with PCA, principal component analysis. What we want to do is that reduce the high dimension into low dimension, but this one can be incorporated in the multi-omics. So you have a global score and try to maximize the covariance within omics and across omics. So it's doable. It's just if you're looking at the mathematical formula, it's very scary if you're not a statistician. So, but these two approaches, we found it's became more mature and more accepted by the community. And the last one is the probability modeling. So there's a lot of things going on here, and it's very beautiful theory. But in practical, it's sometimes working well, sometimes it's not working well, and require quite a lot of computing. So this is something I mentioned about called the multi-view clustering, and we try to use it, and it's working very well. So that's I would like to recommend. What they describe this approach. For example, this example is called spectral clustering. They are developed in computing science. I think it's try to clustering for different. For example, you are a user and you generate a lot of your personal profiles. Like, and then they try to have the different characteristics. Try to describe the single users or stuff. So it's a very natural to use this. They call it a view in this case. So one object from different view, and they basically describe the same thing. And in this case, if we apply to multi-omics, we're talking about the same biological sample. You collect genomics, metabolomics, proteomics. So this is easily transferable. And what they try to do is that they create a similarity graph. And this graph can be aggregated, then have a fused graph, then they do clustering. This one is if you familiar with the SNF, similarity network fusion, and it shares quite a similar concept. So this is very useful from machine learning to multi-omics. So that's one thing we found is quite useful. The other one is dimensionality reduction. This is this has been there quite a long time. So you have the omics, you just basically doing a PCA, but you have a very different constraint. And you have constraint that means you want to maximize not only covariance or variance within each omics, you want to maximize the covariance across omics. So you have an extra score, they call it the global score. So with these constraints, you can extract some factors, latent factors, and then use this condition to gradually find the scores loadings similar to PCA, but it's in a multi-omics context. So this is something it's theoretically working should working well, but practically it's very hard to interpret when you get a result. So it's been there for 10 years, and the application is just start to be more popular. Let's see that. And so we actually did a very thorough survey and try to interpret it in a more plain words, like what's the popular dimension reduction method. And here's that multiple co-inertia analysis, consensus PCA analysis, partial least squares, and procrusting analysis, which is basically just a visualization. Try to put the two omics and try to rotate them and make them just more fit with each other. Because you think there should be some similarity, and you just force that similarity to happen. So a lot of times, such approach became a lot of times it's just a visual things. Whether there's a statistical backup, it's not quite sure, but it's quite often used for several multi-omics. So you just want to see how they are correlated, how they can be best fit into each other. And one of the other ones became common is called Diablo, which is quite a very few multi-omics, and they try to do kind of called a supervised approach. And here I want to say is that the multi-omics is usually give you rich stories about understanding biology, and it's usually not necessarily for the prediction, because we usually get a lot of the features, more than enough features to do a supervised machine learning. If you have multi-omics, and you will get it quite hard. And so most of the multi-omics statistics, they just try to maximize the covariance among these different omics. They don't actually correlate to the final phenotypes. So Diablo is one of the quite rare about supervised approaches. Actually doing well also. So I mentioned about this concept. I hope you more or less follow the overall thinking process. I'm not talking about the very details, because we do all the details every day, try to write a code and do stuff. So I don't want to discuss this. Also, this is not, we don't have enough time. So here is that we have the tools I introduced you for metabolomics and gene expression and microbiome, and we want to integrate with network-based and data-driven. So knowledge-driven and data-driven. We want them to actually talk to each other and try to give better ideas. So this is something that we found that there's a method, but they're not necessarily communicating the result properly. So we want to host them, and through our visual analytic approaches, I discussed it earlier. So this is another tools called MERNet. If you are doing microRNAs, and we have the tools called MERNet. Basically, we have microRNAs related to genes, SNPs, disease, and transcription factors, and put them together in a context. It's very useful. We show a figure on the top right and show that. So this omicsnet is here, omicsnet.ca. We want for multi-omics, but this is knowledge based. We have a very comprehensive knowledge base behind. You can integrate genes, transcription factors, microRNAs, metabolites, microbiome, and the graph. So this is the idea we want people to see. Okay, this is definitely very simplified. We hope it can be like that, but the reality is it's not so easy. Okay, we found a very comprehensive network usually when we apply multiple ones, and it's still so hard to understand. How do we do that? We actually try to detect modules within this whole network. And from each modules, you can actually zoom in and extract them and to further study it. Here we show this microRNAs and their genes correlation networks. So basically, we give them big pictures, but we also allow them to focus on individually to get that. So that's the network-based approach. We basically use people map their significant features to the network and extract them to study them. And here's omics analyst. I tell you, we are working right on this. It's submitted for revision. So we hope we'll be published soon. And so what we try to do is that we have the correlation, we have the clustering. I just mentioned about how do we do a simple correlation analysis, how do we do the multi-view clustering, and how do we do dimensionality reduction. So this one is, there's a method to do that. So there's a lot, but how do you make it into a meaningful things? We need a good visualization and interaction. So we have done this called a heat map and a 3-dimensional scatter plot and a 3-dimensional network. So it's you already see some pictures. So it's quite useful, and we tested in 2 multi-omics like this is proteomics and metabolomics on a recent publication, I think it's Nature Communication, on the human pregnancy. And we see a very clear pattern. We see a very good trajectory. And we put different data, omics, metabolomics, and protein together. When we see their overlap and the pattern is very clear, and biological story just almost screaming. And when we see how they correlated on the network below, correlation network, it's very natural. And we just use search literature and see what you find. It's very easy to come up with a interpretation. So we are very happy with this case study. So once we publish, I hope to get more feedback from other people who actually use the tools to get more things out. So overall, it's a multi-omics. Once you get a good data, good design, and the story, the biology is naturally coming out. So this is something I would like to see. So the one is we talk about future, and I think most time is we focus too much on the algorithms. Now it's a lot of algorithm there, and we want to make it into a translation used by the researchers, by doctors. And so a lot of time is how do we do a better visualization? Nowadays we have the VR, we have the AI. So this is something as a vision and how it's going to happen in 5 years, 10 years. it's there. Okay, it will be there in just sooner or later. So this is something. Finally, it's my my lab, and we are in COVID, so it's mostly working remotely, and I think that's the end of my talk. Thank you.
Great, thank you so much. That was such a wonderful presentation. I myself found it very interesting. Take it to the floor. Does anyone have any questions? Well, that being said, if nothing's jumping up right away, it's kind of, I guess, slightly off topic, but with your tools, I think they really stand out as having a really great user experience. I'm just wondering, do you have a strategy for your approach in when you are developing them to try to ensure that that documentation is there, that people actually can understand how to use your tools?
Yeah, that's a very good question, actually. In order to do that, you need to share the perspective. So to get a good how to say you need to share the perspective of the users or the target who would want to do the data analysis in a certain ways, and you need to share that view, and you need to basically you need to this. If I'm want to do this, how should the other side do? So you usually need to cross the two fields, from a researcher or from a computational or bioinformatics. So you need to have two views to switch back and forth, and you get a happy middle ground. Most time is I realize is the communication across different groups is not as efficient, so that really slow down the translation. So you had a good algorithm, and the other side don't share it or don't quite understand. So this is sometimes also they have such a strong need, and you don't find it interesting. So there's a lot of time as a better communication and share the common goals, and that will be help a lot. And you when you have a tools, and you need to have a collaborator who actually you in your mind ask them to try it, that making sense. They will give you a lot of the comment, and some of them is very cut, and you need to just think about how to address that. So the best one is always you cannot you need to have a balanced view. So yes, I got a hands up, please.
Hey, Jeff, great talk. I had a question about, I guess, the identifying false positives. So I think in one omic, it's very, there's a ton of false positives, right? Just because of the high dimensional nature of it. So do you think that the inclusion of more omics, for lack of a better phrase, kind of cancels out those false positives or exacerbates the issue? Has anyone kind of touched on that? Has anyone kind of done any research into what you actually get out of a multi-omic analysis in regards to false positives?
Yeah, that's an excellent question. I don't think anybody, there's discussion, and there's no good answers towards that. One key reason is that we don't have a very good multi-omics data set for benchmarking analysis. So that's really make it hard to have objective answers to that questions. Overall, in my feeling, is that why I want to emphasize the gradual incremental approach. Also always visualize the data, engage the users, because when you do one, and you have certain ideas, and if the actual layers of omics confirm it, you get more confidence, because that's a I not say it's orthogonal, but actually that's additional evidence to confirm it or refute it. So you do have feelings while you are thinking, gradually collecting more evidence. So so overall is that as long as we're taking a more cautious approach, understand the biology study design, and don't naively believe one or two algorithms give you the magic numbers you need. So it should overall reduce the false positives. So this is something I believe more data will help improve the whole false positive things.
Okay, thank you.
All right, we have a couple more questions. Let's start with William.
Hi, Jeff. That was a great talk. I'm sorry if I missed this, but I know one of your big points was kind of like, if you're thinking about working in a multi-omic space, starting with a single omics data that you know very well, and then kind of adding on additional information from there.
Yeah.
Another way of looking at it from my perspective is if you start from genetic variations, say you're working with like population studies, then like a natural way to branch out from there is starting to map different omics features to the genetic one. And then from there, trying to integrate this information with what we know about different complex traits. So I was kind of wondering what your thoughts were on, so you have like a lot of great tools here for like working with multiple omics outside of genetic variation, but I was wondering what your thoughts are when you actually start looking at genetic variation as like the root.
Oh yeah, you got me. And because I don't, I never deal with the genetic variations. Probably I will. I have a student working on that. So and my field is always a downstream of genomics. So transcriptomics, proteomics, metabolomics. And so I couldn't give you more. Probably you're already the more expert than me. But yeah, I think it's there's no magic bullet. It's always a gentle improvement and respect the current knowns. And so sometimes if you really need a leap of faith to make that connection and you need a good visualization to start to think about that, then you need more validation because that's a leap of faith. So overall is, yeah, it will be easier with more tools and better data. Yeah.
Thanks.
Great, Nafise, would you like to ask your question?
Yeah. Thank you, Jeff. Thank you for your nice presentation. Actually, I want to know where is the position of the epigenetic studies in your diagram? What is Affiliation?
Good question. I think your question and the same to William. I'm in a faculty, we don't deal with epi, we don't deal with genetic variants. So, you know, if I'm working with cancer study, I should be able to answer you. But we are dealing with more different domains. So I don't know. I hope I will be able to answer you soon.
I think, if I can interject, I think like adding DNA methylation as like a layer to this would still fit pretty neatly in the framework that you have for integrating multiple omic layers. That being said, like once you get into like positional information being relevant to how these methods like relate to one another, or sorry, not these methods, these measures relate to one another, then it starts to get a little bit more complicated.
Yeah, I got you. Yes, that part is really, I have student started doing this choral inference from the MGUS, and it's quite tricky. So I think, yeah, we will have some better solution in the near future. And so far, I'm trying to not to touch too much because it's not my funding grant, so I cannot answer that at the moment. But we will start to gradually move on. I believe overall the framework should try to address it, but I don't want to put too hard a limit because clearly there's a position things that you know, you cannot just be, the distance can be in the very other possibilities. So that's when I have no clear answer at the moment. Yeah.
Thank you very much.
Thank you.
All right, well, there are no other questions. I guess we'll call it a day. Thank you so much again, both Betty and Jeff. We really appreciate having you. And
Thank you for having me.
Great end to the Van book season.